about the response of the immune system rather than the ability to enter a cell. Possibly the capsid would be exposed, and if the body had been exposed to that capsid protein before, there might be an immune response. But what about, what do we find in the envelope of envelope viruses?
The spikes that are outside here.
So without the envelope then, the spikes, because this is supposed to be an envelope virus, there won't be any spikes in the capsid. So should this envelope virus be able to enter the cell by it being recognized, by recognizing the host cell receptors? Theoretically not. It is quite feasible that there might be an immune response to this particular virus because the capsid is exposed, and it could well be that your body has already developed an immune response to that capsid protein. However, it does not mean that that viral DNA is completely incapable of causing damage. If there was a DNA-based virus and that DNA is managed by perhaps being piggybacked onto another virus, it may still find its way into the cell and be able to integrate into the host genome. But in general, without its envelope, it doesn't have its spikes, so theoretically, we block the ability to absorb to the cell and carry out its infectious process. Any questions? Okay, so let's move on about viruses. So we've dealt with absorption, so now let's talk about penetration of the virus into the cell. We know it needs to gain entry, which is the second step in replication. How the virus gains entry. How the virus gains entry is really virus specific. So here's the classic way bacteria T4 infects. And by the way, Lambda does basically the same thing. And I showed you this in the EM a few years ago. So the virus lands on its particular receptor. The tail shrinks, and the genetic material is injected through. It actually produces this actual pore by drilling in its tail spikes. So it actually creates this pore that didn't exist before using these tail spikes. And then the DNA is actually injected into the genome of the E. coli. So that's the classic way a bacteriophage gains entry, through this ability to inject the DNA through this pore-like structure that forms as a result of a shrinking of the tail and the tail fibers here engaging in forming a pore. But for animal viruses, it's a little more varied in how they gain entry to animals. So there are several different mechanisms that animal viruses can use. So remember, we're dependent on the initial contact, and remember, random collision is part of it, i.e. there's random collision of cell and viruses, but only those that are capable of absorbing to each other, in essence, will actually lead to an infected cell. That infection is dependent on the presence of the appropriate receptors. And then what can happen with an animal virus is, one, the viral genome can be injected, so similar to what we saw with the bacteriophage. Two, there can be a scenario where the viral capsid enters, and the third one is the entire envelope virus enters. So we'll have injection, an attachment where, as I'll show you in a minute, the envelope remains in the plasma membrane and the capsid gets placed into the cell. And the third one is through a classic endocytotic pathway where the entire virus, including the envelope, enters. So here's two classic viruses that I'll start with, and then I'll go back to more general. So here's HIV. So here's the virus. Here's the envelope. Here's the capsid. Here's the genetic material with the reverse transcriptase attached. That's what the little orange spot is. So the receptors interact with CD4, and there's a second protein that's important in the surface of the mammalian cell, the CCR5. They attach, brings this whole structure in contact with the plasma membrane. There's a fusion between the envelope and the plasma membrane. And then the capsid with the genetic material is basically placed or injected, not really injected, it drops literally into the host cell, and then the capsid actually gets degraded, and then that, remember, HIV is a retrovirus, so it starts as an RNA molecule, it'll be reverse transcribed by the reverse transcriptase into DNA, which ends up in the nucleus. Influenza also is a RNA-based virus with an envelope. It makes use of the sialic acid residues that are present in the viral membrane, attaching to receptors that are sialated, which is a form of glycosylation. So does anybody know what sialic acid residues are? They're actually a specialized form of complex carbohydrates that are made by mammalian cells. Taken biochemistry, you will have learned a bit about these structures. And again, we have an interaction with the receptor, with the particular protein in the envelope. That leads to a receptor-mediated endocytosis. So in order to do receptor-mediated endocytosis, what has to form? We learned in week 10. What kind of specialized pits must form? Coated with platinum. So platinum-coated pits must form. So once this interaction takes place, platinum underlies where the endocytosis will take place. The whole viral particle enters, including the envelope. It enters it is basically sitting in an endosome, which with fusion with the lysosome will lead to low pH. The envelope will actually be removed, and the capsid will also be destroyed by the higher pH, and the genetic material will be released. And the influenza virus, even though it's an RNA virus, also can enter the nucleus. All right. So those are the two examples of viruses showing you the classic model. One, which is the clathrin-mediated endocytosis, which was through influenza. The other, which is the non-endocytic route, which was demonstrated by HIV, where it fuses, where there's a fusion between the membrane of the virus and the membrane of the cell, and then the capsid containing genetic material is then placed into the cell. The capsid will be removed, and then the RNA can be reverse transcribed. So those are the two of the classic routes. The third one would be an actual injection of genetic material, so no entry actually of the capsid. Okay, so animal viruses, can they enter the host cell by receptor-mediated endocytosis? Yes, they can. Can they enter the host by straight endocytosis? I didn't show you a straight endocytosis, but yes, they can. So they don't need clathrin-coated pits to enter. They can enter by a more classical endocytotic pathway. And there is a difference between clathrin-coated endocytosis and straight endocytosis, but the end result is the same. You end up with a mouthful in a endosome that then is damaged by the low pH. Can it enter the host by fusion to the host plasma membrane, a la like HIV? Yes, it can. So in fact, all the above are correct. So animal cells have a lot more variety of ways in which the virus can enter. Now, one of the fundamental differences between animal cells and bacterial cells in terms of their surface is what? What surrounds the plasma membrane of prokaryotic cells? The cell wall. So when the cell wall is present, things like receptor-mediated endocytosis are not going to be feasible because the virus is not engaging on the plasma membrane. The virus is engaging on the cell wall. So the same is true, by the way, of plant viruses. Their genome tends to enter rather than the entire virus. Okay, so once we've attached and we found a way to get the genetic material in, then the virus has to be be replicated, packaged, and it has to exit. So in the case of most viruses, the nucleic acid genome is first released from the nucleocapsid. Okay, so we have to get the capsid out of the way, then the genome is released. If the viral genome is DNA, then that DNA will have to actually make its way to the nucleus to be replicated. And not only will the DNA have to be replicated, but messenger RNA will have to be made. And then that mRNA will have to exit the cell, proteins made, and then for packaging and also for further replication. So the viral genome is then transcribed, then packaged, and then new viruses can leave the cell to infect other cells. Now remember, the plus strand RNA does not need to first be replicated. Remember, the key thing about the plus strand RNA is once it's in the cytoplasm, it can be used to make proteins. It will still have to be replicated into a minus RNA strand to make more genome that is plus RNA, but in terms of making proteins, it does not have to be replicated and transcribed first. Okay, so when I started talking about viruses, I said viruses are non-living because they are dependent on their hosts for survival. They need to hijack the host RNA machinery, protein machinery, DNA replication machinery in order in order for them to make multiple copies of themselves. The only proteins that the genome for most viruses that they have to encode are the specific virus proteins, i.e. the coat proteins, i.e. the spikes that they require on their capsid or in their envelope, as well as encoding the protein that makes the capsid. There are exceptions to that, however. Any kind of virus that undergoes a unique type of transcription or replication that doesn't occur in a host cell will need to bring that with it. So the classic example then would be retroviruses require what enzyme? Reverse transcriptase. So reverse transcriptase not only has to be encoded in the genome, but some of that reverse transcriptase actually has to be packaged in the virion. So this is why in all of these diagrams, by the way, you see the little dot here. What they're saying is reverse transcriptase, the protein is associated with that genetic material. Because in order to start the process, you need some reverse transcriptase present. So that means any specialized polymerase will have to be packaged, made and packaged in the actual viral particle. So if they undergo a specialized movement of plus RNA to minus RNA, then in fact they'll need a specialized protein for that. Otherwise, they use all the host enzymes for the rest of their processes. All right, so how replication takes place is varied depending on the virus. I'm doing 2 hours a bit on viruses. There are whole courses on viruses. So I'm just sort of giving you the highlights about basic viral virology. Okay. If you have an interest in virology, I highly recommend you go take a virology course, because what I'm telling you is a tiny little top of the pyramid, and there's a lot more information about the viruses. But the key point is that in every case in which we require replication, packaging, and export, precursors are required, which means the host has to provide nucleotides for nucleic acid synthesis, ribosomes and amino acids to carry out protein synthesis. So in essence, when a virus enters a cell, basically that cell's sole purpose now is to support the multiplication of that virus. It's not going to be doing most of its normal functions, and therefore hence why we see damage to the cells. There are a few exceptions in terms of packaging of the viral genome where packaging occurs in the DNA, it occurs in the nucleus rather than the cytoplasm. So DNA-based viruses are packaged in the nucleus. Some RNA-based viruses are packaged in the nucleus. The majority of the other viruses are packaged in the cytoplasm. So most RNAs, viruses are actually packaged in the cytoplasm. There are a few exceptions like influenza, which is packaged in the nucleus. So that means once the virus is packaged, if it's packaged into the nucleus, it has to cross more than one membrane. It actually buds off the nuclear membrane. And then what happens is large amounts of virus sort of assemble right at underneath the plasma membrane, and that causes the release. So some will bud out of the plasma membrane, some will bud out of the nuclear membrane, some will actually bud out of the Golgi or the ER. Generally the ones that bud out of the Golgi or the ER, common sense would dictate they were first in the nucleus, and they ended up, because that membrane is contiguous, they end up budding out of the ER or the Golgi. Once, and again, the key point is envelope viruses generally get out of the cell by budding. And keep in mind, the lipid material of an envelope on a virus came from the host. The spikes are encoded by the virus itself. So for example, influenza virus buds from the plasma membrane, herpes virus from the nucleus, or it can also bud from the endoplasmic reticulum. So generally the release of envelope viruses is through classic exocytosis. So a budding and a release to the outside. What has to happen is those viral envelope proteins have to be embedded into the plasma membrane or nuclear membrane of the host. And keep in mind, almost all naked viruses are released via cell lysis. So to understand viral biology, you have to understand cell biology. Because I have just mentioned a whole slew of events that you learned about in cell biology to understand how viruses are moving. So let's just talk about a really simple virus. This is bacteriophage. So the first thing that happens is we inject the DNA, double-stranded DNA, into the host, which is E. coli, so that's infection. That DNA is immediately then used to make early mRNA. And those early mRNA make what's called early proteins. So the early proteins are going to be some of the proteins that are going to be necessary to support phage DNA replication. So phage DNA replication starts when roughly the second level of mRNA is produced. Generally, viruses produce proteins in waves based on how they're needed. So for example, if you're an HIV virus and you enter the cell and you've entered with some reverse transcriptase, that won't be enough to reverse transcribe all of the genome of your virus. So you'll want to make some more reverse transcriptase. So that will be part of your early proteins, or for example, RNA-dependent RNA polymerase may be one of your early proteins. So you have a series of steps of mRNA and proteins leading to your proteins that make your head, your tail, and your collar or base plate. And all of these self-assemble. So they are not assembled by other proteins. They self-assemble into the structure. And then once the head is actually formed, the genetic material enters. And this is true of all viral particles. So the capsid forms before the capsid doesn't form around the genetic material. The capsid preassembles, and then the genetic material goes into the capsid. Okay, so just before the break, let's discuss two important pathways of viruses: the lytic cycle and the lysogenic cycle. So lytic would tell you, what does the lytic cycle do? It causes the host cell to lyse. The lysogenic pathway is really what we call the pathway where the virus becomes quiescent. It becomes quiet, it gets integrated into the host chromosome, and it is not actively involved in causing an infection. So bacteriophages, as well as several other viruses, have the ability to both undergo a lytic pathway and a lysogenic pathway. So the lytic pathway is what we've already talked about. Viral genome enters, we have replication, we have packaging, we have release of the phage particles. In the lysogenic pathway, the viral DNA gets integrated into the host chromosome. It sits there as a prophase. It will be replicated every time the host chromosome gets replicated, so it will be passed on to the progeny of that host cell. As I said, this in itself will not cause a viral infection, but could cause damage to the host DNA. But at a certain point in time, with the right kind of signal, these prophases can reemerge, come out of the chromosomal DNA, and turn back into a lytic viral infection. So we actually look for lytic infections caused by viruses. They form what's called plaques. So this is the plaques you will see when E. coli gets infected by T4 or lambda-phage. So can everybody see here, there's actually a background of bacteria and there's clearing in a nice circle. So there's no bacteria that are in these circles. Each one of those are a plaque. Each one represents an E. coli cell that's undergone multiple rounds of replication. So in the world of microbiology, we define a bacterial colony as a colony that has been given rise to by what? A single cell. So a plaque is defined as being given rise to by the infection of a single phage particle by a single cell. And then once that cell releases viral particles, it goes and affects more cells, and you end up with a clearing. So you can count colonies on a plate, or you can count plaques on a plate, and determine the original titer of bacterial cells or viral particles. And you will do this in the lab. So the way in which this works is you start with a nutrient agar plate, you add your, and this is remember this is a bacteriophage, so we have the host, which is E. coli, we add the virus, we allow them to hang out together for a little while, the phage attaches, it infects the E. coli cells, and then we put that mix into what's called a mixture that we originally put onto the surface of the bacteria. So, sorry, the surface of the plate. So what we have in this tube is a molten top agar. So it's liquefied agar to which we've added E. coli cells and a bacteriophage. We pour it onto the surface of the plate, and most of the bacteria remain uninfected, so we get a lawn of bacteria. But wherever we see a plaque, that represents an E. coli cell that's been infected by a phage. You will do this in lab. At the same time you do a generation time lab where you look at the growth of E. coli, you will also do an infection with bacteria. So after the break, we'll start talking about how these viral particles get out of the cell. So it's 10 after, by that clock, so come back at 20 after.